where he was like?
Short answer is no, because Dr. Cumming never went through them to see which questions are good or bad. So
Got it. Okay.
at the moment, no, but we will also try to avoid those types of questions in the coming exam.
Okay, that's
So,
totally fine.
I mean, we can talk about the coming up and see if she wants to go through this.
Okay. Okay, thank you.
Good morning. There's lots of stuff to get through. All the stuff we talked about today will be on the on the test in 2 days. So I don't want to rush too much, but I'd like to get through most of it. So we're going to get going. The exciting world of microtubule motor proteins. Okay, so last time we talked about kinesin. Kinesin is the plus end directed motor. Plus N is anterograde transport. Kinesin is made up of basically 2 heavy chains, and it's the heavy chains that have a head, neck, and tail domain. And the head domain binds to microtubule. The head domain uses ATP. With kinesin, it moves to the plus end. And now we're moving on to a protein called dynein. Dynein is the minus end-directed motor that moves on to microtubule, so it's moving retrograde, so basically away from the cell surface, usually. Okay, so it's moving towards the minus end of the microtubules. So, as I mentioned, most motor proteins have very similar domains. They have heavy chains, so we talked about kinesin heavy chains, we're now talking about dynein heavy chains, and later on we'll talk about myosin heavy chains. They all have the same structure, a head domain, a neck domain, and a tail domain, although they're slightly different names. So don't let the naming confuse you, it's the structures that are more important, especially this head domain. Okay, so again, the heavy chains tend to work in pairs. So with kinesin, you had 2 heavy chains, which are the 2 feet that moved along when it looks like it's walking. And now with dynein, you also have 2 heavy chains that are working together. Okay, these heavy chains have head domains, neck domains, and tail domains. Just artistically, they are drawn differently, and the names are slightly different, but they're still basically the same type of protein. Right? There's a head domain, a neck domain that's flexible, and this tail domain that eventually will lead to cargo. They're called slightly different things here. Here, it's called a stem, but it's basically a tail. right? Again, the head domain, so this is sort of a structural representation of the protein. In red is the head domain, this linker is the neck domain, and this stem is basically the tail domain. The head domain is large, the head domain is what binds to the microtubule, the head domain is what uses ATP to move to the minus end. There's another structure inside the head domain that's called the stalk. So when you look at this binding to microtubules, it looks sort of different. But again, that's sort of artistic representation. It doesn't matter what it looks like. The head domain is binding to the microtubule, there's a neck domain, and then there's this tail domain that's going to be binding to cargo. Okay, so dynein has two heavy chains, and the two heavy chains are walking towards the minus end, where the head of the heavy chain is using ATP. With kinesin, it had light chains to bind to cargo. With dynein, it uses many different proteins to bind to cargo, and these proteins form this complex called a dynactin complex. Sometimes it's called a dynactin heterocomplex because there's different types of proteins that are involved that bind to different types of cargo. So with kinesin, you have different light chains. With dynein, you have different proteins in this dynactin heterocomplex. These proteins are going to bind to these tail domains of the dynein heavy chains. So here is the dynein heavy chain, there's a head domain, a neck domain, and this tail-like structure, right? So you have many proteins bind to this tail domain, and then it's binding to the cargo. So different types of dynactin heterocomplex can bind to different types of cargoes. And they're going to be pulled towards the minus end by the two dynein heavy chains. Make sense? A couple of differences here between dynein and kinesin is that we sort of understand a little bit of the mechanism of cargo release. So dynein is going to move to the minus end, carrying some sort of cargo. When it gets to the minus end of the microtubule, it has to release the proteins, release the cargo. And we know that there's a protein called dynamitin that's involved in that. So when the cargo gets to the minus end, dynamitin basically explodes the complex, hence the name. So it kind of causes the complex to fall apart and the cargo is released. So cargo release is important, and in dynein you sort of understand a little bit of that mechanism with this protein called dynamitin. Another thing that we see here that's a little different than kinesin is that there's this other protein called P150 glued that's also bound to the microtubule. So P150 glued doesn't use energy, and it doesn't really, it's not involved in powering anything, it's not helping movement, it's just helping to stabilize things. So as the dynein heavy chain, the heads are moving to the minus end, P150 glued is sort of behind it, binding to the microtubule. You can almost think of this as sort of like a railway carriage, right? You have the engine, so at the end here, you have the engine, which is the dynein heavy chains, and then behind it is sort of like another train car being pulled along, and this train car helps stabilize all this cargo. So P150 glue binds to the microtubule as well as the dynein heavy chain does, except that P150 glued isn't propelling or using any energy, it's just sort of stabilizing things as they move along towards the minus end. All right, so you have kinesin involved in antegrade transport towards the plus end, dynein involved in retrograde transport toward the minus end of the microtubules. And just to kind of remind you, therefore, processes that are going to go antegrade or retrograde are going to involve these proteins. So there's a little figure here trying to show you things moving towards the centrosome and away from the centrosome, and there's arrows trying to show you movement. I'm not sure what these arrows are representing. It doesn't really matter what they're representing. If you want to go towards the minus end, you're going to use dynein. If you want to move towards the plus end, you're going to use kinesin. Okay? More importantly, though, what's useful on this figure is to show you that as things are being transported, the motor proteins themselves can be cargo. So if you have some sort of pigment vesicle here being transported, you know, as it's moving towards the plus end or towards the minus end, you can actually have the other protein as cargo being moved along. So if you have kinesin taking some sort of vesicle to the plus end of the microtubule, potentially you're going to need dynein to bring something else back. How do you get the dynein to the plus end so it can move back to the minus end? Well, the dynein itself can be the cargo that's part of the transport. So they're trying to show you here a lot of these structures, for example, this mitochondria here has both dynein and kinesin on it, so that that mitochondria can move both towards the minus end and towards the plus end. So it's also important to remember that the motor proteins themselves can be part of cargo to be moved to the plus end to the minus end so that they're in the right location for the transport. Another thing that's sort of on this slide that's not quite related to that is to point out that the movement of some of these motor proteins is influenced by the stability of the microtubules. And recently it was found out that the alpha subunit, I think it says that somewhere here, is stabilized. So if you acetylate the alpha subunit, it stabilizes the microtubules. So not only can MAP stabilize microtubules, but acetylation of the alpha subunit can stabilize this, and this acetylation also promotes kinesin 1 movement. So kinesin 1 will move faster along the microtubule that's been acetylated versus one that hasn't been. Okay, and again, just to kind of hammer home this idea that you have heavy chains, and the heavy chains are the important molecules. So both kinesin and dynein have two heavy chains that often work together for transport, right? So the two heavy chains have similar structures, although they have slightly different names. The important part is both a kinesin heavy chain or a dynein heavy chain, they both have a head domain. It's that head domain that binds to the microtubule. It's the head domain that's the ATPase. They both have a neck-like linker region that's flexible. They both have a long stalk that's basically a tail. That tail is called different things. Sometimes it's called a stalk, sometimes it's called a stem, but it's basically a tail. I will always call these things head, neck, and tail. So I think it makes sense. Right? The fact that dynein has this extra structure inside its head, fine, but really it's still the head domain that's binding to the microtubule. Okay, so kinesin and dynein are the motor proteins in microtubules. They are involved in vesicular transport or anything that needs to be transported along microtubules from the plus end to the minus end, lots of vesicles and other structures. However, there's also very specialized functions of microtubules. So we're going to talk about two very specialized functions of microtubules, and that's cilia and flagellar beating and mitosis. So let's talk about microtubule function in cilia and flagella. Right, so cilia and flagella are basically two versions of the same thing. The only difference is one is short and one is long. So flagella is long, cilia is short. It's sort of a gradient. I don't think there's a common definition of when does a cilia become a flagella as it gets longer. It's just that it's short in general, relatively short, it's a cilia, and flagella tends to be long. Also, cells can have lots of cilia, but they tend to have few flagella. Okay, so cilia are short, flagella are long. Both of them are undergoing bending. They bend, and when they bend, they move things. So they move the liquid that's around them. So either they will move liquid, so the cell will stay stationary, and the cilia that's beating is propelling liquid. So you have cilia, for example, in your lungs that are constantly beating, getting dust out of your lungs. Or things like flagella can propel cells. So you have sperm cells that have flagella on them, and the cell is moving. So cilia and flagella are moving things, so they're either moving things across the cell surface or they're moving the cell itself. And they do so by bending. And the structure that they are bending is called the axoneme. So the tail of a cilia or flagella, if you do a cross-section of the tail, you see this structure that's called an axoneme. So the cilia and flagella cross-section looks identical. It's made up of many proteins. More than 250 proteins are found in cilia and flagella. Most of them, we don't know what they're doing. They're probably involved in regulating the bending, which we'll talk about. What you do see inside cilia and flagella are rings of doublet microtubules. Okay, so in this example, there's 9 doublet microtubules in a ring. So if you remember, a doublet microtubule has an A tubule of 13 protofilaments and a B tubule of 10 protofilaments. Okay, these doublet microtubules are stable. So once you make a cilia or flagella, it's basically stable. It doesn't tend to polymerize or depolymerize. So axoneme has 9 doublet microtubules. And it also may have singlet microtubules in the middle. In this case, it has 2 singlet microtubules. Those singlet microtubules are also stable. Okay, these are not cytoplasmic singlet microtubules, these are axonemal singlet microtubules, so all of these microtubules are stable. So this has 9 doublets and 2 singlets, so this is called a 9 plus 2. Most cells, most animals have 9 plus 2, but there are other conformations of this where you might have 1 singlet microtubule or no, or you have different number of doublet microtubules on the outside. But most of what we see are 9 plus 2. Okay, there's a lot of proteins in here. A couple of them I'd like you to memorize. One is this protein called nexin. So nexin is a sort of blue ring that holds all these doublet microtubules in a ring. So nexin is stabilizing things. Nexin links all the doublet microtubules and keeps things stable. There's also a protein called a radial spokehead. So from all of the doublets, you have these other proteins called radial spokeheads that are also stabilizing things. They're holding things in place. So you have 9 doublet microtubules that are being held in place by nexin and these radial spokeheads. Additionally, coming off of each of those doublets, off of each of those A tubules, is dynein, but this is axonemal dynein. So this dynein is different because its tail, it's permanently bound to the A tubule. So the tail of the dynein heavy chain is permanently bound to the A tubule, and the head of the heavy chain is reaching towards the neighboring B tubule. So you have the motor protein dynein, but it's axonemal dynein, so it's not doing transport like it normally does. It's actually physically attached to the A tubule, the tail of it is, while the head of it is reaching towards the neighbor's B tubule. And you'll see how that works in a second. Okay, so you have this 9 plus 2 arrangement of a cilia and a flagella. Microtubules come from a microtubule organizing center, and when you look at the base of the cilia and the flagella, you see this structure called the basal body. So the basal body is the microtubule organizing center of cilia and flagella. And when you look at the structure of the basal body, you see that it's made up of triplet microtubules. So the basal body has 9 triplet microtubules. If you remember, a triplet microtubule has an A tubule of 10 protofilaments and a B tubule of, an A tubule of 13 protofilaments, a B tubule of 10, and a C tubule of 10. Okay, so the basal body has 9 triplet microtubules, and during the creation of the cilia and the flagella, this acts as the nucleating center, and you get polymerization of these microtubules, and they polymerize towards the cell surface and beyond the cell surface, except you go through this thing called a transition zone at the cell surface. So at that transition zone, you switch from triplet microtubule to doublet microtubules. So you lose the C tubule. So the basal body is triplet microtubules having A, B, C tubules. And then through that transition zone, you lose that C tubule, so now you have doublet microtubules. So you're going from 33 protofilaments to 23 protofilaments. Also, in that transition zone, you either gain or lose these singlet microtubules that are in the center. There doesn't seem to be a rule as to sort of what happens in these singlet microtubules. So in this example, this basal body has 1 singlet microtubule in the middle. In the transition zone, you lose that singlet microtubule, but then in the axoneme, in the cilia and flagella, you get 2 singlet microtubules. We don't know why those singlet microtubules are there, what they're doing, but they are there. Okay, so the basal body is the microtubule organizing center for ciliary flagella. It's made up of triplet microtubules, but as they polymerize in the cilia and flagella, you lose that C-tubule, so you only have doublet microtubules in the cilia and flagella. When you look at some cells that have cilia and flagella, often the cilia and flagella come out of the cell surface in pairs. So here you can see a cilia flagella coming off in pairs, and there are two basal bodies sitting that are acting as the microtubule organizing center for these two cilia and flagella. So you have these two triplet microtubule structures that are basically lying 90 degrees to each other, which is very similar to centrioles. The basal bodies and centrioles are very similar to each other. The basal bodies are polymerizing the microtubules in cilia and flagella. The centrioles are found in centrosomes, but the centrioles don't directly polymerize microtubules. But here the basal bodies do directly polymerize microtubules. Make sense? All right, so now the more complicated part, axonemal bending. So the axoneme is the structure. You have this 9+2 arrangement of doublet microtubules, and it's going to cause the bending of the cilia and flagella to cause motion, basically. So this bending is generated by dynein. There's dynein, that's axonemal dynein. That dynein is bound, the dynein tail is bound to the A tubule, and the head reaches towards the B tubule. The textbook has messed up this diagram overly in a bunch of ways, so don't look at too many of the labels on this diagram, just sort of the idea of what should be happening. For example, here it has a B tubule labeled, and that's clearly an A tubule because it's a whole structure. So don't look at what's an A tubule, never mind where the heads are, just that there is dynein. The dynein is attached to one one doublet, I should say. The dynein is attached to one dimer, and the head is reaching the other doublet. Okay? So, you have the head domain reaching and binding to the next microtubule. Dynein is a -n directed motor. So, when the head domain binds to this other microtubule, it's going to want to move to the -n. So when that head domain tries to move to that -n, you're going to cause tension going on. Okay, so what's going to happen? So let's look at an experimental situation, first of all. Inside the axoneme, there is nexin present. So in between these doublets, nexin protein is present. Okay, so this is showing you that there's nexin. So inside an actual cilia flagella, there's nexin present that's going to restrict movement. But experimentally, let's remove nexin. So let's look at an experiment where there is no nexin holding these doublets together. If there is no nexin holding these doublets together, and you have axonemal dynein between them, the head domain is going to bind to one of the doublets, and the head domain is going to try to move to the -n. Right? So as the head domain tries to move to the -n, there's going to be relative motion of these doublet microtubules. That relative motion will cause sliding. So there's arrows trying to show you helpfully what's moving where. That may or may not be helpful. Just picture in your mind, you have this head domain that's bound on this doublet microtubule. This head domain is going to want to move to this -n. So this head domain is going to move to the -n. So the distance between this head domain and this -n will decrease because this head domain will move towards it. So as that distance decreases, that top doublet microtubule is going to move to the right. Okay? So this green arrow is showing you the movement of the top microtubule, while the head is actually moving to the left. So the head is trying to move to the left, the doublet microtubule moves to the right. So there is sliding that occurs. So that if there is no nexin, you get relative microtubule sliding. Hopefully that makes sense. However, nexin is present. You can't slide. There's these proteins holding things together. So you have these dynein heads trying to move to the -n. but they're restricted because nexin is restricting things. So the best way I can try to describe this is to try to use my hands as sort of examples, right? So if you can imagine one hand is a doublet microtubule, another hand is another doublet microtubule, and I have dining on the tip of my fingers. The bottom of my palm is the minus n. So if I have dining on the tip of my fingers and I bind to another microtubule, the dining on the tip of this finger is going to want to move to the minus n here. If there is no nexin, it will. It'll just move down and you have sliding. So if there's no nexin, the dining, you're just going to move them and you slide. However, if there's nexin, you can't slide. So imagine you have nexin gluing your palms together. So now you try to move the tip of your fingers down your other hand. When you try to move the tip of your fingers down, but your palms are stuck, you're going to end up bending. Okay, so bending results because sliding is restricted. Even if you can't picture that with my palms or your palms or whatever, it doesn't matter. Bending is occurring because sliding is restricted. Okay, so dining is moving, there's relative motion of those doublet microtubules, and the restriction due to dining is going to cause sliding. It's going to cause bending. Hopefully people get that. A couple things to kind of complicate that. Let's go back to this. Okay? Everything can't slide all at once. That's the backup slide. When you look at a cilia or flagella, there's bending in different regions at different times. So this bending isn't all binding all at once and everything is sliding at once. It's localized. There's only localized bending in different parts of the axoneme, and it's usually a wave sort of structure. So when you're considering this sort of configuration, you have dining here that can bind to this doublet microtubule, this dining can bind to this one, this dining can bind to this one. If all those dinings bind all at once and all of them try to move all at once, this thing would just blow itself apart because everything is trying to move all at the same time. That doesn't happen. Bending is only localized. Right? That's why you get this sort of sigmoidal sort of structure, this wave of things. So bending is localized, and probably some of those other 250 proteins inside this axoneme are controlling where the bending occurs and how it occurs. Okay, bending is occurring because of dining, but that bending is restricted because of nexin, and therefore you get the bending. Good, hopefully? Okay, so cilia and flagella, this bending action allows them to propel things. However, cilia and flagella are also crucial in signaling, right? Because they're basically like antennas sticking out there. There's these long structures sticking away from the cell. So cilia are often, and flagella often have receptors on them, and there's all kinds of molecules being sent to the tip of the cilia and back down towards the cell body. So there is much transport that occurs within the axoneme. So in the addition to having axonemal dining, inside the cilia, you also have kinesin and cytoplasmic dining because you're undergoing antegrade and retrograde transport within the cilia or the flagella. So you can still transport things to the plus end away from the cell body and then to the minus end. So there is still transport going on, and using the motor proteins we sort of talked about, cytoplasmic dining, and in this case it's kinesin 2. Make sense? So you can still transport in cilia and flagella. And this signaling is particularly important during embryogenesis. A couple of decades ago, people realized that many cells have something called a non-motile cilia. So it's a non-motile primary cilia. So lots of cells have these cilia, and it looks like it has an axonemal structure, but it's not a functioning axoneme because there is no axonemal dining. These structures can't bend. But what they can do is they can be involved in signaling. So we're not quite sure how important this signaling is sort of in adult tissues, but in embryonic tissues, this primary cilia, this primary non-motile cilia, is crucial in development for receiving signals. So Almost all embryonic cells have this primary non-motile cilia, and they are involved in signaling. If you get rid of this primary non-motile cilia in embryos, you get embryonic consequences. So lots of signaling pathways. Sonic hedgehog, for example, involves these localization of things to these non-motile cilia. Okay, so cilia and flagella are involved in bending, moving things. They can also be involved in signaling, and this signaling is particularly important during embryogenesis with these structures called non-motile cilia that can't bend, but you can still transport things, and they're involved in signaling. Okay, so that was somewhat complex, but an example of how how microtubules have a specific role in cilia and flagella function. And now moving along quickly, but I'll try not to go too quickly, is now looking at microtubule function in mitosis. Okay, you guys have learned mitosis, all kinds of courses before, I'm sure, anaphase, metaphase, telophase, whatever, cytokinesis. You know all these different stages from other things, but now we're going to look at them in the context of the cytoskeleton. Okay, so mitosis is actually sort of two separate things. It's karyokinesis, which is the separation of the chromosomes, and that involves microtubules, which we'll talk about now, and cytokinesis, which is the division of the cytoplasm, and that involves actin, which we'll talk about when we talk about actin. So when we're looking through all these different stages now, interphase, prophase, metaphase, anaphase, etc., we're looking at specifically the role of the microtubules. Okay, so what are the microtubules doing to allow karyokinesis, which is the separation of those chromosomes? So those chromosomes have duplicated, you've replicated your DNA, now you have to separate that DNA. And it's the microtubules that do that. Okay, so this sort of pattern is sort of a general pattern where not all cells do all of these things. Different cells have different sort of lengths where they do all these different stages. You know, one of the first things is nuclear membrane breakdown. And some cells don't break down their nucleus. Some yeast cells still have a nucleus all the way through this. So these are sort of just general rules of what the cytoskeleton is doing. Okay, there's always exceptions in biology. All right, so one of the first things that has to happen is that you have to replicate your centrosomes. So the centrosome is the microtubule organizing center in most cells. The centrosome is going to be the spindle poles, and it's going to be responsible for organizing the structure called the mitotic apparatus. So you have to replicate the centrioles. Okay, so the centrioles are replicated. So once the centrioles are replicated, the centrosome is basically no longer a centrosome because it has different characteristics. So if I'm using the word centrosome, it's the microtubule organizing center in a non-mitotic cell, and it has certain characteristics. When it replicates, even though it still has centrioles and looks similar, it's still a microtubule organizing center. It's no longer called the centrosome. It's a different microtubule organizing center called the spindle pole. Okay, it's the pole of the mitotic apparatus, and that's because it has different characteristics to the microtubules that it's polymerizing. So if you look at a non-mitotic cell and look at microtubules, they have a certain characteristic. They're dynamically unstable, but they're relatively stable. And that's because of the characteristics given to them by the centrosome. So during interphase, or when it's non-mitotic, the half-life of a microtubule might be quite long. It could be minutes, 5 minutes, for example, there. So the centrosome is polymerizing microtubules. They're relatively stable, even though they're dynamically unstable. During mitosis, that's very different. Right? So during mitosis, the centrosome is replicating. When the centrosome is replicating, all the non-mitotic microtubules depolymerize, and then they're repolymerized again to form this mitotic apparatus. When they're repolymerized, their half-life is very different. Their half-life is in seconds now. So the microtubules coming from the spindle pole are growing and shrinking very quickly. And that's important to their function because their function is to capture and move chromosomes. And you want to do that quickly. So so the two microtubule organizing centers, the centrosome has a very different function. It's involved in transport of organelles, etc. But once it replicates, it becomes a spindle pole. Its job is very different. Its job is to capture chromosomes and segregate them as fast as possible before mistakes happen. Okay, so this stability is due to a number of things, but one of them is related to the stability of maps and map utilization, right? So when you look at why are mitotic microtubules so unstable, one of the reasons is you look at map stability, especially in this map called XMAP215. So there's certain things that can depolymerize microtubules, such as kinesin 13. So if you remember, kinesin 13 uses ATP to remove dimers. So it's not that microtubules are unstable because there's an increase of kinesin 13 activity. Microtubules are unstable is because the maps stabilizing them are basically removed. Okay, so there's much less map activity, and therefore during mitosis, these microtubules are much less stable. All right, the centrosome is replicated. You separate the centrosome, you depolymerize all the microtubules, and now they're going to repolymerize, and eventually they're going to form the structure called the mitotic apparatus. So the mitotic apparatus has these two spindle poles. They used to be the centrosome, but they're no longer the centrosome because their characteristics are different. So the spindle pole is polymerizing microtubules. All the rules we've learned so far still hold. The minus end of the microtubules is at the microtubule organizing center, and the plus end is away from the microtubule organizing center. So now the microtubules are spreading away from these poles, this microtubule organizing center, and they're forming this structure. And based on where the microtubules are going, they are named different things. So there's polar microtubules, kinetochore microtubules, and astral microtubules. Okay? So the names are pretty self-explanatory to some extent. If a microtubule leaves the pole and then goes and binds to a chromosome at the kinetochore, it's a kinetochore microtubule. So kinetochore microtubule links the pole to the kinetochore of the chromosome. If a microtubule is polymerizing and it's moving towards the other pole, but it misses the chromosome, it's kind of reaching towards the other pole, it's a polar microtubule. So polar microtubules are kind of reaching pole to pole, but they're missing the chromosomes. But you end up overlapping polar microtubules. So polar microtubules, there's a lot of them, they're just overlapping, but they're not binding to the chromosomes. The polar microtubule and the kinetochore microtubule together are called the spindle. So the spindle is all the microtubules in between the two poles. So pole to pole is the spindle microtubules, and that's the polar microtubule and the kinetochore microtubule. There are other microtubules that are polymerizing that are going away from the other pole. They're sort of going behind things, if you like. So these microtubules that are not polymerizing toward the chromosomes, not polymerizing towards the other chromosomes, people thought they looked like a star, so they're called the astral microtubules. So astral microtubules are microtubules that are going away, polymerizing away from the other pole. Okay, so you have astral microtubules, which together with the spindle microtubules form the mitotic apparatus, where the spindle microtubules are the polar microtubules and the kinetochore microtubules. Easy, huh? All right, so the kinetochore. The kinetochore is a protein structure that's found in an area of the chromosome called the centromere. Okay, so different centromeres have, different chromosomes have centromeres in different positions. They all have this structure called the kinetochore. The kinetochore is made up of hundreds of proteins. So these proteins are where these microtubules are going to bind to, and if a microtubule binds to the kinetochore, it becomes a kinetochore microtubule. Make sense? So microtubule is growing, it's the plus end of the microtubule that's growing towards the chromosome. When it hits the kinetochore, the microtubule will be attached to the proteins within the kinetochore that will grab onto the microtubule. When you look at the kinetochore, you can see layers in it, and it's often described as an inner kinetochore and an outer kinetochore. And that's important because when the microtubule is captured, the plus end of the microtubule is still free. So when a microtubule is growing from the pole, it hits the kinetochore with the plus end. The kinetochore grabs that microtubule, but that plus end is still free. The plus end is not capped. The plus end can still polymerize and depolymerize. And it needs to polymerize and depolymerize because when it does so, it can move the chromosome back and forth towards the pole or away from the pole, depending on the direction it needs to go. Right? There's a movie that hopefully will work that's sort of going to show you the overview of what's going to happen, and then I'll talk through it after this. Okay, let's hope this works. Hold on, I'll do this again. Okay, everybody quiet. Dynamic microtubules radiating from spindle poles attach to chromosomes at the kinetochore and align them at the equator of the cell. Spindle microtubules rapidly fluctuate between growing and shrinking until a chromosome is captured at its kinetochore. Taking a closer look, the plus ends of multiple microtubules attach to the outer plane of the kinetochore. Once captured, a combination of two mechanisms are thought to generate the force to move the chromosome. First, polymerization and depolymerization at the plus end that attached microtubules can push or pull the chromosome. In addition, a plus end directed motor proteins such as cont'd is known to push the kinetochore away from the pole along the growing microtubule. A minus end directed motor protein such as dynein could pull the kinetochore toward the pole, while cont'd tethers the kinetochore to the shrinking microtubule. All right, so kind of going through that a little bit again. So you have microtubules coming from both sides going towards the kinetochore, and once it binds to the kinetochore, the plus end is still free. So the plus end can polymerize and depolymerize. At the kinetochore are many proteins, including kinesin and dynein. So in this little diagram, there's a whole bunch of different kinesins drawn here and various dyneins. Kind of ignore the numbers of the kinesin. Kinesin is a plus end directed motor, dynein is a minus end directed motor. Okay, so you have the plus ends of the chromosomes at the kinetochore, but those plus ends are free unlike it's drawn here. Right? The microtubules are held away from the plus end. So on both sides you have kinesin and dynein, and on both sides you can polymerize and depolymerize the microtubules. Right. So if you imagine, if you want to move the chromosome to the right, you would depolymerize the microtubules on this one side, and if you want to move to the minus end, you need dynein. So dynein is going to move to the minus end, and you're going to pull the chromosome to the right. On this side, you can polymerize the plus end on this side, and you can use kinesin to move to the plus side to push the chromosome. So you're pulling on one side, pushing on the other side. Okay, so this way you can capture chromosomes from both sides and then move them to the metaphase plate. Okay, so that involves polymerization and depolymerization at the plus end, and also kinesin and dynein on both sides. Hopefully that makes sense. The other thing that's important is you have to capture all of the chromosomes, and you have to align all of the chromosomes at the metaphase plate. There's all kinds of checkpoints here so that you don't start anything until everything is lined up at the metaphase plate. Then you can start separating things. So there's a lot of checkpoints that are involved here. We know a lot of them how this works. One of them is, how do you know that the chromosome is captured from both sides? You don't want a microtubule coming and binding this sister pair here, and if there's nothing on the other side, you just pull it to the right, and then therefore you haven't separated the sister chromatids. So how do you know that things are captured from both sides? Right? So you need tension for this. So to assure biorientation, that is to say that there's microtubules grabbing this chromosome from both sides so that when you separate, you're separating the sister chromatid. You use tension. So within the kinetochore proteins is this protein called NDC80. So NDC80 will grab on to the microtubule, but it'll hold on for a little bit. Okay? So you have a microtubule coming in, it's going to bind to the outer kinetochore proteins, and it's going to hold that chromosome. So now the cell is waiting for another microtubule to capture this chromosome from the other side. When it does that, there will be tension. Both the microtubules will pull, and there will be tension. So if you capture the chromosome from both sides, there will be tension. If there is no tension, that means you've only captured it from one side. So if there is no tension, a kinase called Aurora B phosphorylates this protein called NDC80. So if you don't capture it from the other side, there is no tension. Aurora B phosphorylates NDC80, which is part of this kinetochore, and NDC80 lets go of the microtubule. So in the presence of tension, Aurora B does not phosphorylate things, and therefore NDC80 holds on to both sides, and it's all good. Then later you can move the chromosome to the metaphase plate and separate. Okay, so you need tension, you need you need microtubules from both sides. In the presence of tension, Aurora A is not active. If there is no tension, Aurora A is active, and it will phosphorylate NDC80, which will let go of the microtubule. I know it's fast, but hopefully that all makes sense. Okay, so this ensures that you've captured all of the chromosomes from both sides. If you don't capture them from both sides, the chromosome will start to be pulled one way and it'll be released. And the cell is monitoring all this. It somehow knows that all the chromosome pairs have been captured. So you've captured...
Sorry, would they have to capture it at exactly the same time?
They don't have to capture them at the same time. They can be captured at different times. Eventually, they all need to be lined up at the metaphase plate, though. So they can be captured at different times, but they have to be captured from both sides. Once they're captured from both sides, then they all have to be lined up in the metaphase plate. The metaphase plate is sort of the center, and then they're going to be moving away from that metaphase plate during anaphase. Why? Because cytokinesis is going to come in through there. So you want things to move away from this cytokinesis occurring so that all the chromosomes are segregated before you divide up the cytoplasm. So you want to start off in the middle because you want to move away from the middle because that's where cytokinesis is going to occur. So you've captured the chromosomes from both sides. You have these spindle poles that have organized this. You have the kinetochore microtubules that line up all of the chromosomes. But now you need to move this spindle into the center. You need to line it up with the metaphase plate. And here you use these cortical anchors that are found on both sides. So you have these astral microtubules, if you remember. These astral microtubules are reaching away from the mitotic apparatus. They're reaching the plasma membrane. Within that plasma membrane is dynein. So dynein, if you remember, is a -N directed motor. The -N is at the pole. So these astral microtubules are attached to the plasma membrane dynein, and now, because dynein is there, you can move this whole mitotic apparatus either to the right or to the left, depending on which way it needs to go. And you do that by activating either one side of this cortical anchor or the other side. In this case, because you want to move things to the right, you would activate the dynein on this right side. So the dynein is going to be bound to these astral microtubules. The dynein is a -N directed motor. The -N is sitting in this spindle pole So the dyne is going to pull on these microtubules and then pull this pole to the right, and therefore you're pulling the whole mitotic apparatus to the right. So dyne is sitting on the plasma membrane and pulling on those astromicrotubules. You can imagine as dyne is pulling on these astromicrotubules, you have to depolymerize these astromicrotubules, otherwise as you're pulling on them, you're going to poke them right through the plasma membrane. So dyne is pulling on the astromicrotubules, and those astromicrotubules are shrinking. All right, so you've captured the chromosomes from both sides of the kinetochores, and then you line them up in the metaphase plate, and then once those checkpoints are met, then you're going to separate all these chromosomes. Okay, so this is busy, but hopefully it'll make sense. Okay, there are two things going on here, right? There's anaphase A and anaphase B, right? So anaphase A is moving the chromosome closer to the pole. Okay, so in anaphase A, the kinetochore microtubules are shrinking. Right, the kinetochore microtubule moves is from the pole to the kinetochore. If you remember, at the kinetochore, you have all kinds of motor proteins, right? Specifically, you have kinesins and dyneins. So as this kinetochore microtubule is shrinking, the distance between the chromosomes and the pole is shrinking. So anaphase A involves the depolymerization of the molecule of the kinetochore microtubules, and therefore the distance between the chromosome and the spindle is decreasing. Right? So you're separating the sister chromatids because of depolymerization of those kinetochore microtubules. Make sense? While this is occurring, anaphase B is also occurring. Anaphase B is pushing the poles apart. So in one part, the chromosome is moving towards the pole, and the other part, the poles are moving apart. Why is this occurring? Well, you have these polar microtubules that overlap. In between those polar microtubules is kinesin 5. Kinesin 5 is a plus N directed motor. Those microtubules are overlapping. Kinesin 5, as it tries to move towards the plus end of both these polar microtubules, it causes those polar microtubules to unoverlap. It undoes that overlap and pushes those polar microtubules apart, and therefore pushes the poles apart. Okay, so complex, but hopefully easy enough to remember from that first line, or first little blurb up at the top. Anaphase A involves microtubules shortening, and that decreases the distance from the chromosome to the pole, and anaphase B involves kinesin 5 undoing overlap of the polar microtubules, and that pushes the poles apart. That's the level you need to know that too. All right. Good luck Friday.
Hi sir, it's a quick question. Is P150 a part of the tail?
Pardon me?
Is P150 a part of the tail?
No. It's another protein part of the dynactin mineral. It's part of the dynactin mineral complex.
Okay. Thank you.
Because I already know that they have different move directions when they do the complex.
Yep.
The problem is why it is the dining is not
You mean the cilia and flagella? Or do you mean the tail of the protein? So no, that's so in the axoneme, dynein is used to move microtubules. Why is it not kinesin? I don't know. It's just the way it's dynein. That's evolution. I don't know why.
just part of
Joining is what's responsible for exiting the venue.
Light. You hear Sonic on that song?
Lights.
Nah.